Hi everyone, welcome to my talk. I'm happy to be here today and present to you part of my master's project, where I looked at the effects of mercury over behavioral plasticity in an Arctic seabird. This is a project done in collaboration with researchers from McGill University and Environment Canada, and it's part of a bigger NCP project. The Arctic is warming twice the global average, phenomena that has been linked to changes in sea ice coverage, such as early sea ice breakup and melting. Timing of sea ice breakup is a key factor in ecology of seasonally ice-covered waters. For example, sea ice breakup initiates phytoplankton bloom, increasing food availability for upper trophic levels. In the case of seabirds, these animals tend to match chick rearing with these food availability peaks. However, previous studies have shown that although some species are breeding earlier, they are still not able to adjust their breeding to match the environmental changes. Moreover, other studies have shown shifts in primary productivity, leading to changes in diet and foraging behavior, modifications that could potentially increase the cost of reproduction and reduce adult survival. In addition to climate change, seabirds in the Arctic are also affected by chemical contamination. Mercury is one of the contaminants of concern in this region, where it bioaccumulates and biomagnifies in the food web. This contaminant is a potent neurotoxic as well as an endocrine disruptor. To this date, most studies have focused on the impact of chemical contaminants over behaviors such as laying date, nest attendance, and chick feeding rates. The assumption is that high levels of these behaviors are associated with fitness. However, there is growing awareness that fitness is poorly associated with individual values of any of these behaviors, and rather fitness is associated with plasticity in these traits. So on this project, what we wanted to explore was if mercury in fact is affecting behavioral plasticity. The way animals respond to environmental changes is highly regulated by hormones. As I briefly mentioned in one of my previous slides, chemical contaminants like mercury can alter the endocrine function, affecting the way wildlife responds to changes in their environment. For this specific study, we focus on thyroid and corticosterone hormones. Thyroid hormones are involved in multiple physiological traits and are known to regulate metabolism. Specifically, T3 and T4, which are tridotinoin and thyroxine, regulate tissue oxygen consumption and thermoregulation. In seabirds, T3 is also associated with basal metabolic rate. Specifically for diving seabirds, what this means is that high T3 levels can increase basal metabolic rate and therefore reduce the time seabirds spend diving. Whereas corticosterone is a stress response hormone that triggers physiological and behavioral adjustments during stressful events. In the context of climate change, it is particularly important for wildlife to be able to rapidly respond to environmental modifications. In this study, we wanted to explore if chemical contaminants, specifically mercury, is affecting seabirds' ability to adapt to climate change in the Arctic. We particularly focus on the potential relationships between circulating mercury concentrations with circulating thyroid and corticosterone levels, and with foraging behaviors. And we hypothesize that mercury disrupts behavioral plasticity in response to changes in ice availability by altering hormone levels. In this research, we studied the thick-billed murre, which is a deep-diving seabird widely distributed in the Arctic. This species is associated with ice and is sensitive to changes in environmental conditions such as temperature. We conducted this study at Coates Island in Hudson Bay, where up to 30,000 pairs of these birds nest every year on its cliffs. First, we looked at the associations between hormone concentrations and mercury using linear models. What we found is that total mercury was correlated with total T3. This association was significant in 2016 and 2017, but not in 2018. In 2016, as you can see in the plot in the left, total T3 increased with total mercury concentration. As you can see in the plot in the middle, in 2017, we found the opposite trend where total T3 levels decreased with total mercury concentration. And we looked at the other associations between mercury and the rest of the hormones. However, none of the other associations appeared to be significant. Next, we looked at the associations between hormone concentration and each of the foraging behaviors using linear models. What we found is that total T3 was significantly correlated with the proportion of time the murres spent diving. We found this relationship to be significant in 2016 and 2017, but not 2018. As you can see in the first two plots, we found that the proportion of time spent diving decreased as total T3 levels increased. However, it is important to highlight that this relationship stopped being significant for 2016, as you can see in this p-value that I'm reporting on the first plot, once I remove two outliers. However, we do still think this relationship is real and biologically relevant, although our model might not be indicating that it's significant given the small sample size for this year. We did found other significant relationships between the different hormones and other foraging behaviors. However, for the sake of time, I won't really mention them on this talk. In summary, we found that total mercury is affecting circulating T3, and T3 is correlated with total time spent diving. We think that mercury might be affecting total T3 at brain level or at the thyroid gland level, causing a potential thyroid function disruption. Moreover, total T3 was negatively correlated with total time spent underwater in 2017 and potentially in 2016 as well. Therefore, we think that in warm years like 2016 and 2017, mercury might be disrupting total T3 levels and thus interfering with how murres can adjust to changing ice levels by altering their diving behavior, something that we didn't see in 2018, which was a year with normal or better ice conditions, where we think that in normal conditions, the murres might be able to compensate the effects of these contaminants and their effect over their endocrine function. Finally, I just wanted to thank everyone involved in this project, including the people that helped even before I joined. And thank you all for listening. I'll be happy to take any questions.